but nothing at the bottom.
Okay,
well, I have all the icons across the bottom. I have record on the bottom of mine.
Okay, so Kurt, you should be the host at this point, and we're recording.
Okay.
All right. Okay, so good evening, everybody, and thanks for joining us. My name is Tara Schroeder. I'm the Education Coordinator for Green Mountain Conservation Group, and if you're just joining us, welcome. If you haven't heard of Green Mountain Conservation Group, we're located in the Ossipee watershed region of New Hampshire, and we're a small nonprofit watershed organization that focuses on protection of natural resources and in our region of New Hampshire. So that includes, you know, the forests, the land, the water, the wildlife, and so on and so forth. And so we're really pleased tonight to welcome Kurt Laffin from who's a wildlife biologist, and he's going to be providing us with this wonderful program about the restoration of the American chestnut tree. And I also want to extend a thanks to our board member, Mark Longley, who's also a member of the American Chestnut Foundation, for introducing us to Kurt and this suggestion for a program on the American chestnut tree. I know there are a lot of folks out there who are very interested in this topic. So I'm interested myself in learning more about the history, and I'm hopeful for the recovery of this wonderful piece of our history. And without further ado, I'm going to hand it over to Kurt. And if folks have questions throughout the program, we will get to those. You can use your chat feature, which is located at the bottom center of your screen, or it should be there. And just go ahead and type in your question, and then we'll read them out to Kurt at the end of the program, and he can answer your questions. All right, Kurt, it's all yours.
Okay, well, thank you very much, Tara and Mark, for arranging this. It's my first attempt at doing one of these online, but I think it's going to work out just fine. One question I often get is, how did I ever get interested in the American chestnut tree? Well, when I was about 5 years old, my father took me to a huge, beautiful tree in Harvard, Massachusetts, and he told me that was the last living American chestnut tree that he was aware of. And I've always had an interest in natural resources. I pursued a career in wildlife science, and I kept seeing these references to how important the American chestnut tree had been to the eastern United States. So about 12 years ago, when I found out there was an organization trying to bring this tree back, I just had to get involved. So, and I've heard that story from several other people. It's even my wife, Carol, who used to go to her family farm out in Pennsylvania, has a similar story. She was told about the American chestnut tree. So what we're going to do this evening is spend a little bit of time trying to convince you how important this tree was, and then we'll get into the bad news about how we almost lost it, and then the good news about how the American Chestnut Foundation and other groups are working pretty diligently to try to bring it back. So the first thing we want to clear up is that we are not talking about the horse chestnut tree. We're talking about the American chestnut tree. The horse chestnut tree was introduced into the United States way back in the 1700s from Europe. It's an exotic tree. In fact, there's one growing in Portsmouth, New Hampshire that was planted when the Declaration of Independence was signed. It's at the Moffett Ladd Historic House in Portsmouth. The horse chestnut tree is it's an ornamental. Fortunately, it will not survive in our forests. It does produce a burr and a single nut in each burr. But we are talking about the American chestnut tree, which is native to the eastern United States. Used to be a massive tree that grew straight and tall, had all kinds of benefits. Its flowering structure is very small. When you do see one of the leaf is similar to a beech tree because they are related. But the flowering structures are nothing like the horse chestnut. These little green things are the female flowers that eventually become the burrs. And when you do see one of these flowering, what you will see are these catkins that produce the pollen, and the little female flowers that become the burrs of these little structures right in that area. The burrs are about the size of a tennis ball when they mature, and if it's fully pollinated, it will produce 3 nuts in each burr. The historic range extended all the way from Maine right down through Georgia and west out to the Mississippi River. We know they were here in Massachusetts because these 2 guys, who you might recognize, showed me and my wife Carol the remnants of an American chestnut tree. All you can see is a little bit of the arc of the tree. So that tree was probably about 6 feet in diameter. Of course, you recognize Mark and Fred Levine. They showed me this. It's right at the Meade Base Camp on the south side of the White Mountains National Forest near Sandwich. So we certainly know they were here in New Hampshire. They had tremendous cultural and economic benefits. As you already saw, they were very large trees. They grew very fast. They resisted rot. The lumber was straight and did not split. It was used for many, many things. And remarkably, it was a food source. Native Americans certainly knew the value of these trees and used them for many different purposes. The early settlers, of course, used what was at hand, and a lot of that material was American chestnut. If you ride through the Blue Ridge Parkway down in North Carolina, you'll see miles of these old chestnut fences that have been there for well over 100 years. This utility pole is down at Cape Cod. It was put up in 1928, and it's still there, still functioning. I mentioned that the high tannin content. Tannin was used to make leather. The wood would be chipped and boiled and melted down to a liquor, and then the hides would be exposed to the tannin, and the tannin would replace the water in the hides to the process of making leather. This mill in Compton, North Carolina, used over 100,000 cords of chestnut every year just to extract tannin. And incidentally, some of those of you who are old enough to remember when your grammar school teacher had a big green ink blotter on her desk, someone figured out that after the tannin was extracted, the pulp could be used to make a low-grade paper that was very absorbent. And that was used to make those ink blotters. I mean, I'm old enough to remember those ink blotters, but maybe some of you aren't. It was because it was used for furniture, because it was a very readily available and good wood. This bed happens to be in the Robert Frost house in Derry, New Hampshire. The U.S. Forest Service keeps records of what different woods are used in each state every year. And then I found this report from 1912, and it listed these various furniture items and whatever. And when I'm doing my talk in person, I ask if anybody in the audience knows what hames are. And very few people do, because we don't live on farms anymore where we have draft horses. But this is a set of hames. And what's interesting is that there was a mill in Sunapee, New Hampshire, that produced so many hames made with chestnut that the Forest Service added that to their annual report. And a remarkable thing about this tree, the nuts are an amazing food source, both for people and wildlife. Wagon loads of chestnuts used to be brought into cities on the East Coast and sold on the street as roasted chestnuts. And for wildlife and livestock, John Eckley did this sketch showing what wildlife species used chestnut, but I don't think he won any art contests, but he certainly made his point that it was very important to wildlife. Unfortunately, we weren't able to really document how important it was or how it was a part of wildlife ecology, because we didn't really start studying wildlife populations and wildlife management until the mid-1930s. And by then, the American chestnut was pretty much on the way out. Well, today, none of these benefits are still there. all these benefits are gone. The chestnut tree is essentially functionally extinct. It's still here with us, but it just cannot be used for any of these purposes. Well, what went wrong? Around 1904, the New York Botanical Garden and around New York City started realizing that their chestnut trees were dying at a remarkable rate. And they weren't sure what was going on, but eventually they figured out that it was a blight, Cryphonectris parasitica, chestnut blight that was imported to the United States with an Asian or Chinese chestnut tree. It's an airborne fungus and it spread very quickly. By the 1950s, it's estimated that it killed about 4 billion trees across 200 million acres of the eastern United States. And the way blight works, one of those spores enters a little fissure or crack in the bark, and then it eventually grows around the circumference of the tree and literally strangles it. But what it does not kill the roots. It kills the tree from the point of infection upstream, up to the top of the tree. So keep that little fact in mind of how the blight works, because we'll get back to that later. The blight spread very quickly. You can see in 1904, it showed up in New York. By 1920, it had pretty much consumed southern New England. And then by the 1950s, it had spread all across the range of the chestnut tree because it was an airborne fungus. Well, you can imagine to having lost such a resource that everything that people could do was tried to stop the blight and save the tree. There was some crossing between the Chinese and the American chestnut, but those trees were just about as vulnerable as the wild trees. So it was very ineffective. So you might say, well, if the Asian tree could resist the blight, why not just let the Asian tree keep going? Well, the problem with that is the Asian tree, this is a mature Chinese chestnut tree, and you can see it does produce nuts that can be eaten, but the tree just doesn't grow anything like the American chestnut So enough bad news. Let's get into some good news. Dr. Charles Burnham was a plant geneticist from the University of Minnesota who spent his career using a process called genetic backcrossing to produce agricultural crops that could resist various diseases. And after he retired, he got together with one of his cohorts, Phil Rutter and a few other people, and they decided, well, let's try this genetic backcrossing with American chestnuts and see if we can bring them back. So they created the American Chestnut Foundation in 1983. So the American Chestnut Foundation works under 3 different approaches to try to restore the American chestnut tree. And we call it the 3 birds. And there's biological control, backcross breeding, and biotechnology. Now, biological control is a process that can be used to slow down the impact of chestnut blight on individual trees. This is used in Europe, where the European chestnut tree is in large orchards that produce nuts. If you go in the grocery store around Thanksgiving time and buy chestnuts, they're probably European chestnuts, Castanea sativa. Well, they use a process called hypovirulence, where they inject a fungus into chestnut blight cankers that actually slow down the growth of the canker. Another process that can be done is very simple, called mud packing. Now remember earlier I pointed out that the roots of the tree don't kill. There's something going on in the soils that can resist chestnut blight. So if an individual tree has a canker on it, you can literally take a handful of mud, moisten it, open up the wound a little bit, put that mud right on the canker, wrap it in plastic, and sometimes that will actually slow down the growth. But of course, that's just not a practical way to bring a tree back to a total forest environment. So the next process is backcross breeding. This is the process that Dr. Burnham started back in 1983. There are just millions, probably hundreds of millions of chestnuts still growing all through the historic range, but most of them just get up to 2 to 3 inches in diameter, and the blight kills them. But occasionally, that some of these trees will live long enough to flower. That's what we call mother trees. And the backcross breeding process was to find mother trees and start genetically backcrossing them with the Chinese tree to try to develop a tree that could resist the blight. This is a rather complicated chart, but don't worry, you're not going to be tested on it. If you start the backcrossing process, first the American and the Chinese would be crossed, and that would produce an F1 generation, and it would be pollinated with American chestnut pollen. So now we got a tree that's half American and half Chinese. Well, then the offspring of that tree would again be crossed with American chestnut pollen. You got a tree that's 3/4 American. And you keep going down the list, we get down to the third generation, the fourth generation, and you actually are starting to intercross the backcross trees with each other. So at each generation, the intent was for these trees to retain the American characteristics, but also inherit the genes needed to resist the blight. And it was thought at that time that there were only 3 or 4 genes that were needed from the Chinese tree to resist the blight. At each of those generations, the trees would be intentionally inoculated with chestnut blight, and most of them would die. But the point was that you would keep the very few that showed the most resistant to the blight, and they would be used to go on to the next generation. That was the backcross breeding approach. And this process was developed at the Chestnut Foundation's Glen Place Research Farm in Meadowview, Virginia. That's where it all started. Well, we're talking about a 200 million acre area that where we want to restore this tree. How do you get it all across the range? Well, this is what Meadowview looked like a few years ago. You could still see some of the remaining chestnut trees there. But this is a little 160 acre farm in Virginia. How do we get it spread across 200 million acres? Well, you create chapters in each of the states within the range and have those chapters duplicate the process that was developed at Meadowview. Here's our Vermont, New Hampshire chapter up here. All the different states do have chapters, and they have gone through the backcross process. That involves controlled pollination. We would find mother trees, go to those trees just as the flowers were developing, cut the catkins away, put the flowers in little bags so they wouldn't get pollinated with anything else, come back in about a week when the flowers are opening and hand pollinate them, put them back in the bag, and then go develop or go collect the burrs after they had developed. That was the backcross breeding program. Our Vermont, New Hampshire chapter has these trees growing in 11 different orchards. Our chapter only began in 2007. Our neighboring chapters, such as Maine and Massachusetts, Rhode Island, have been doing this a lot longer than we, and they have many more orchards going through this process than we do within our 11 orchards. Well, this is what an orchard looks like when they are first planted. This happens to be at the Beaverbrook Conservation Area in Hollis, New Hampshire. Well, you've probably heard this one. The more you know, the more you don't know. Well, we know a lot about chestnut trees, and within the last 2 or 3 years, our geneticists have come to the realization that the breeding program is a lot more complicated than originally thought. Instead of 3 or 4 genes required from the Chinese tree to resist the blight, it's probably a lot more than that. So that complicates things because we have to breed it down to a lot more, or we're going to have a tree that's not as American as we want it to be. We're still continuing with the breeding program, but fortunately, we have another technique. This is the biotechnology approach. And we get into the term transgenics. Now, transgenics means this is a process of introducing genes from an outside organism, from one organism into another, with the intent of enabling the latter to exhibit the new property that can be transmitted to its offspring. That's okay. I'll give you a second or two to absorb that concept. That we're trying to change, it's genetic engineering. We're trying to introduce something into the chestnut tree that can be transmitted to its offspring that will make the chestnut tree tolerate the blight. That's transgenesis. The New York chapter of the American Chestnut Foundation, when this whole process of backcross breeding started back in 1983, decided it was going to take a whole different approach. And there's a gentleman named Herb Darling, and he was a New York gentleman who was interested in chestnut restoration, and he teamed up with the State University of New York in Syracuse, the College of Environmental Science and Forestry, and they started to try to create a tree that could tolerate chestnut blight in the laboratory. The tree they produced is called the Darling tree. Darling 54, now it's Darling 58. That's a strange term. It's not just a darling tree, it's a herb darling tree that started the whole process. That can be kind of complicated. This is a picture of the saplings from wild Americans, the darling tree, and the Chinese tree that had been exposed to chestnut blight. And you can see the darling tree is doing quite well. What the College of Environmental Science and Forestry figured out after 30 years of trying, they were able to extract 2 genes from wheat and move those genes into an American chestnut tree. My grandson is a geneticist, and he told me that the genome of wheat is much, much more complicated than the genome of a human being, which is rather remarkable. But when you think about the fact that some of these plants have been around for 1 billion or more years, and we haven't been around nearly that long, that helps you understand how complicated genetic can become. That something that we think is as simple as wheat is much more complicated than we are. Well, anyway, by inserting these 2 genes into the American chestnut tree, it produced an enzyme called oxalate oxidase. And the tree, the American chestnut tree that has the oxalate oxidase is still almost a pure American and shows as much resistance or tolerance of the blight as the Chinese tree. And as we go back to the definition of transgenesis, that about half of the trees produced through this process inherit the oxalate oxidase. Now, oxalate oxidase is nothing to be afraid of. It's in many things that we eat every day. Potatoes and beets and bananas and black tea and dark chocolate. It's a very common enzyme in our environment. So by putting this oxalate oxidase into a chestnut tree, it's certainly not creating anything that's foreign to the environment. And the gene is very widespread, but it's not in chestnut. So the College of Environmental Science and Forestry has been able to insert oxalate oxidase into a chestnut tree. Well, what does that mean? What happens? Well, when the blight attacks a chestnut tree, it does two things. It first produces oxalic acid, and that oxalic acid starts to kill the cells within the cambium layer right under the bark of the chestnut tree, and then that allows the invading fungus to come along and actually kill the tree. So what oxalate oxidase does is detoxifies oxalic acid. It simply neutralizes it into hydrogen peroxide and carbon dioxide. So the blight is still there, but the oxalic oxidase has neutralized the oxalic acid, so the cells of the tree can tolerate the blight. It doesn't kill anything. It simply detoxifies the oxalic acid. All right, we're talking about a genetically modified organism. That means it has to be approved by several federal agencies. The U.S. Department of Agriculture, EPA, and the Food and Drug Administration. Right now, the approval process is going through the U.S. Department of Agriculture. In fact, the comment period opened last month and will be open until October 19th. So if you have an opinion on this whole process and you want to comment on it, you can go to the Chestnut Foundation webpage, acf.org, and you will see a link to public comment period. Now, if you go there, you will see how to make your comment, but you will also see the 300-page application that the College of Environmental Science and Forestry submitted to the U.S. Department of Agriculture, and it will answer just about any question you might have about the whole process. So that process is ongoing right now. Now, here's a complication. The transgenic tree is a clone. That means it has no genetic diversity. Every transgenic tree is like every other transgenic tree. It's as if every person looked like every other person. So we need to interject genetic diversity into the chestnut tree population if we expect to bring the tree back into our forest. So to fill this void, in other words, to create the genetic diversity we need, the Chestnut Foundation and certainly our chapter are currently doing everything it can to find wild trees that are still out there in our forest that are producing viable nuts. And in fact, we're coming up on the harvest season pretty soon. This is an interesting season. We thought we had 2 good trees coming ahead in the Lee, New Hampshire area, but we checked those the other day, and because it's been so dry, the burrs are just dropping prematurely and not producing anything. But anyway, that's just an off season. That happens all the time in nature. When we do collect these nuts, we plant them in what we call germplasm conservation orchards. This is to preserve the germplasm, the genetic makeup of these different trees. So we will, if the transgenic tree is approved and we're able to move forward with it, the trees in the pure American chestnuts growing in these germplasm conservation orchards will be pollinated with the transgenic tree pollen, and then that will help us start to develop this genetically diverse population that we need. If you happen to know where there is a nut producing tree, we certainly want to know about it. You can contact our regional science coordinator, that's Kendra Collins. She's up in Burlington, Vermont, with the U.S. Forest Service Experiment Station up there. And if you go to our webpage, again, acf.org, you'll find this locator form where you fill out the information and then you send a leaf sample along with the form to Kendra. There is also an app you can put on your cell phone called Tree Snap, and if you get it, you will see that right in Tree Snap there different species of trees show up. So you find American chestnut, you take a picture of the tree, and then you answer a variety of questions, and that automatically goes into the system. So this is really crucial that we find as many of these nut producing trees as we can. If you want to get involved in the whole process, we'd certainly love to have you come aboard as a member. If I was talking to you in person, I would be handing out a brochure, but you can go online and become a member of the Chestnut Foundation. And when you join the Chestnut Foundation, you automatically become a member of the chapter of whatever state you're in. And there are volunteer opportunities. Right now, our big one is locating these nut-producing chestnut trees. You can volunteer at an orchard, organize educational plantings, or even invite your local organizations to learn about chestnut restoration by contacting the Chestnut Foundation, or you could contact me directly. And then we can, I don't know if we're ever going to get back to doing in-person presentations, but this seems to be working pretty well. So that's pretty much the story. We're working on all three of the approaches, the biocontrol, the backcross breeding, and the biotechnology. And we're still optimistic. We're not about to give up. We think this can be done, and we're pretty excited about it. So our main contacts are Kendra up at the New England Science Coordinator, and Doug McLean, he's our chapter president. He's in Plymouth, New Hampshire. And incidentally, one of the questions we always get is, how can I get a chestnut tree? Well, Doug has we have 3 germplasm conservation orchards going. We have one up in Burlington, Vermont. We just started one in Rutland, Vermont. And Doug has one that he has on his property in Plymouth, New Hampshire. And right now, he has some extra seedlings. These are pure American chestnuts, so they will definitely get the blight. But if you would like to get a couple of chestnut saplings and plant them, you should contact Doug. And there is his email and phone number, if you can, how you can contact him. But if you want to get some of these, you're going to have to move pretty fast, because you've got to get them in the ground pretty quick with the colder weather coming on board. So that's pretty much the story.
Kurt, can you hear me? Kurt, is my voice working?
I hear somebody.
Okay, it's Doug McLean, and Kurt, I apologize. I actually just got on and heard you say my name. So yes, we will have trees available for sure in the spring. And fact, I have a pretty good number right now that folk could get before the ground freezes.
So, okay, there you go.
Any questions?
All right.
And Kurt, I will post, this is Tara, I'll post that information on the Green Mountain Conservation Group Facebook page and our website too, for
Great.
folks
Wonderful.
that are looking for saplings.
Okay.
And I
Yep.
think we have one question so far. If anyone else has questions, go ahead and use your chat feature, which is at the bottom center of your screen. The question we have from Aaron is, how do I tell if I have American chestnuts? I have huge rows of chestnuts. And maybe you already, spoke to that when you kind of differentiated in the beginning between the horse chestnut and the American chestnut. But any telltale ways that you can tell the difference?
Okay. Was the question, can you tell the difference between a horse chestnut and American chestnut? Was that the question?
It sounds like she has chestnuts, but she wants to know if they're American chestnuts or horse chestnuts or any other type.
Okay. It gets a little complicated. You can go to the webpage, acf.org. All right. If it's a horse chestnut, they are very easy to distinguish between and the American or Chinese chestnuts, because they have a large compound leaf. There are 5 leaflets on each leaf. And the burrs, you can pick up a burr from a horse chestnut tree and handle it very easily. Now, if it's an American chestnut or a Chinese chestnut, and there are plenty of Chinese chestnuts around, people have planted them. Almost everyone you find, though, is going to be out in the open. It's going to be out in full sunlight on a school campus or along on a residential roadway. The Chinese tree and the American tree look quite similar, but the leaf is different. The leaf, one little trick we use, if it looks like a canoe, it's probably an American chestnut. If it looks like a rowboat, it's probably a Chinese chestnut. And if you look at it with a good hand lens, you look on the underside of the leaf, if you see any little tiny hairs on the leaf, it's not going to be an American chestnut. It's probably a Chinese chestnut. And the leaf is going to be more shiny. Now, to complicate things even more, there's a European chestnut. We were just over in Lee a couple of weeks ago checking on a tree, and I'm pretty sure it's a European tree. The leaf looks a lot like the American. It's longer and narrow, but it's kind of shiny. And I just stood back and looked at that tree, and there was just something un-American about it. It just didn't come across as an American chestnut. I did collect a leaf sample, and I've sent it up to Kendra for analysis. We'll see what she figures out. But anyway, I don't know.
So is it possible, Kurt, for people to send a leaf to Kendra to get it identified for sure?
Yes. Go back again.
Okay.
Go back to the webpage.
For Aaron.
Get that locator form. Now, we do keep a record of every American chestnut tree we find. Kendra maintains a huge database of all these trees that we have found. And so when you look at the locator form, it asks you for coordinates and how accessible is it, and are there burrs on it, any signs of blight, things like that. So yes, okay. That's how to do it.
All right, well, we'll move on to the next question from Joanne. She says, what has happened with the tree at the hospice house? And I'm not familiar with the hospice house, but maybe you can answer that.
That sounds like Joanne Dowd. That's a great question, Joanne. One thing I didn't clarify, in our search for finding pure American chestnut nuts, you don't have to just find a tree that's producing nuts. If we're aware of 2 known American chestnuts, we can cross-pollinate them with each other. And if this is the Joanne I think it is, she found a tree at the hospice house in Merrimack, New Hampshire, right on the entrance road. And then there was another tree out in Mount Vernon, New Hampshire, about 10 miles away. And in the spring, early summer, we went out and collected pollen from both of those trees and pollinated the other tree. So we're getting close to harvest time. Probably in the next week or two, and Joanne, I'll let you know about this, we will go out and collect the burrs that we cross-pollinated with hopes that we've got 2 new sources. So yes, Joanne, we're very much on top of that tree, but I still think we're a week or 2 away from harvest time.
Okay, so the next one is from Mark. He says, thanks for inviting me to the American Chestnut Program. What happens, I'm working on a project for sugar maple research, so I can't come tonight. My question is how to winterize the baby tree. What should I put around its tender stem to protect it from mice? It isn't even big enough yet for one of those white wraparound apple tree protections. So maybe some mouse poison. I don't want to build a nest for mice with mulch or compost. Okay. And also, can I post a link of the recording of this webinar to be posted on the GMCG website? And yes, I can do that.
Okay, Carol, did you get that question?
Kurt, I might have some input on that question.
Go ahead, Doc.
Okay, I would say the best bet is using hardware cloth. So if you just get hardware cloth, it can either be the 1/4-inch mesh or the whatever the smaller mesh is. And cut a piece about 2 feet long and 10 inches high and wrap it around the tree and jiggle it back and forth so it goes into the ground, a 1/2 inch or 1 inch. That will pretty much protect it from mice or voles, underground things. Not a bad idea to put a little piece of hardware cloth across the top to keep a possible squirrel out, but I've had very, very little winter predation. We planted 110 chestnuts a year ago this spring and did not lose any depredation. I think a crow pulled 1 or 2 up right after we planted them, but you want to keep your mulch, whether it's hardwood chips or leaves or whatever, away from the stem at all times. There should be about 1 inch or 2 of bare ground around the tree to discourage animals from burrowing. But I don't think it doesn't need anything special for the winter other than it's a good idea to put some hardware cloth around it. That would be my idea.
Okay, thanks, Doug. Eric has a question. He says, assuming that the transgenic tree is approved, how far away do you think a blight-resistant tree is? 1 year, 5 years?
That's a great question. Doug, you might be able to answer this better than I can, but we're probably still maybe as much as 2 years away from approval, because we're just going through the USDA comment period right now, and we still have to deal with EPA and the Food and Drug Administration. So, but let's assume it's approved within 2 years. If we have our germplasm conservation orchards up and running, and enough of those trees survive to flower, then we can be pollinating maybe in the next 3 years. And then we have to let those nuts get planted in orchards. So there's another several years. So it's hard to say, but it's still years away. Doug, do you have anything to add to that?
Yeah, I might be a little more optimistic on the timeline in that there will actually be transgenic nuts harvested here in just a couple few weeks, although without a federal permit, they cannot be distributed. They can be planted in a few approved locations, but there's actually going to be a surplus of nuts that will probably have to be destroyed, waiting for federal approval. But I guess what I was going to get at is that once federal approval comes, if it came in 2 years, there could be nuts available the following year, maybe not for anyone that wants them, but probably for people who have put any time in working for the American Chestnut Foundation. Anyways, I think it could be as quickly as three years from now.
Okay, thanks, Doug. Thanks, Kurt. So Erin follows up about her chestnuts. She was wondering what species they were, and she says they're probably European. Thanks, Kurt. Mark says, Kurt, can one see the comments posted on the Fed's public comment section? Any trends? Question mark.
I didn't get that question. Can you?
Sure, I'll reread it. So Mark asked, Kurt, can one see the comments posted on the Fed's public comment section? I'm assuming this is about the patent for the transgender or the transgenic tree. Yeah. Any trends, he's asking.
So,
is there a public comment section on the federal page about the transgenic tree patent?
Kurt, I can cover that.
Go ahead.
All the comments are very much open to anyone. You simply, I don't know if Kurt's put up the site or mentioned it, but whatever the site is, as soon as you go there, you can read through. There are, I believe it's over 2,000 comments now. Wow. For a while, they were trending very positive. Then the anti-TG tree folks got their act together, and they've rather been flooding the comments. But I think it ebbs and flows. Certainly, the majority of comments are positive. This is a little bit of a biased opinion, but the anti-comments tend to all fit in a fairly small mold. There's not a lot of new information anybody throws out. But I would highly encourage anyone to read them. You wouldn't want to read 2,000, but just scan through them. They're quite interesting.
All right. We have a question from Allison Leach. She says, we have a 75-foot American chestnut tree that is in our front yard that is producing some viable nuts. Roger Willoughby from the Maine chapter and Brian Ross from UMaine have both come and taken samples, and Mr. Ross was successful in grafting cuttings from us. We also have several on our property that have produced viable nuts. So that's great news. Thanks for sharing that, Allison.
Where are these trees? In Maine?
It must be in Maine. She doesn't say.
Yeah. Well, if
maybe she'll follow up, possibly.
Yeah, he's apparently in touch with the Maine chapter if Brian Roth is involved, and
yeah,
and that sounds like a good tree.
Yeah. Okay, we have a few more here. Let me see if we can get through them. Let's see. Mark is asking, let's see, if a cross between the pure ACT and transgenic variety is not called a hybrid, what is it to be called?
I'm not. Doug, any help there?
Yeah, that's an interesting piece of semantics. The scientific answer would be a transgenic tree will be 99.999% pure American. So that's pretty close to 100%. The hybrid trees that the foundation has been working with tend to be about 93% pure American. The transgenic is 99.999% pure American. One gene different out of 35,000 genes. something.
Okay, so we have another question coming in about mud packing, the mud packing method. Is the mud packing method practical for helping a tree survive to the point of flowering so that it could be useful for your program? I have a tree about 3 inches in diameter that has shown blight this year.
Okay, Doug could probably elaborate on this, but I'd say certainly mud pack it. And sometimes it works, sometimes it doesn't. I live in Hudson, New Hampshire, right next to what used to be Benson's Wild Animal Farm. It's now a town park. I planted some of the breeding program trees down there 10 or 11 years ago. One of them has been doing remarkably well. It's about 6 inches in diameter, about 30 feet high. And it had shown no signs of blight until this year, and a little bit started to show up. So my wife Carol and I went down and we mud packed it, and we'll see what happens. But if you don't mud pack it, it's a pretty sure chance you're going to lose it. Doug, you might want to elaborate on that. We've done a lot of mud packing.
I actually have recently. I've probably done 6 or 8 this year. 2 years ago, when we were out visiting the ESF in Syracuse, New York, which is where the transgenic tree was developed, they've done quite a lot of mud packing on some fairly large trees, and there's no question but what it works. Don't be intimidated, just do it. It's simple. I almost love it. It's like when I used to help a mason, and they'd say, go mix up a batch of mud. And this is really the same thing. It's, instead of mixing up the cement, just pour in get some dirt. I grab a little from the compost pile, a little bit from the potato patch, get a good variety that'll have good microorganisms and fungal hyphae. Mix it up with a little water. Take a knife and cut out some of the dead wood on the wounded part of the tree. I forget, what's the word, Kurt? Not the callus, the
Canker.
The canker. Cut out some of the dead wood on the canker so that you expose some of the live wood around the edge. You can very quickly see it's white or green and looks live. And put the mud on it. Sometimes I put mud all the way around the tree, wrap it with saran wrap, and take a lot of masking tape and tape it right up. I've taken off several different mud packings, and there's no question about what it starts healing in from the edge of the wound.
Great information. Thanks, Doug. One more question here from Mark. What is involved in the federal approval process for the transgenic tree? And then he follows up with a second question. Does the American Chestnut Foundation have an annual meeting?
Okay, I'll answer the second part first. The American Chestnut Foundation has had an annual meeting up until this year, of course, because of the COVID. And my wife Carol and I have been to quite a few of them. And I can tell you, if you are interested in American chestnut restoration, you want to go to one of these meetings. Just the energy among the people and find out what's going on and how different chapters are interacting. It really gets you pumped up. As far as the approval, the other part was about the approval process of the question.
Yes, what is involved in the federal approval process for the transgenic tree?
Okay, well, Doug, again, you can probably elaborate on this, but the different agencies have different responsibilities. The question before the U.S. Department of Agriculture is, should this genetically modified organism be regulated, or is it safe enough to let it be distributed? An amazing thing about this is that almost all the other requests for approval of a genetically modified organism are organisms that have to be controlled to keep them from spreading. Well, with this situation, it's just the reverse. We want this spread around 200 million acres of the eastern United States. So they're having some interesting discussions about how to deal with this. Now, with EPA, they first wanted to treat it as a pesticide. Well, it doesn't kill anything. The oxalic, the oxidase does not kill anything. It's not a pesticide. So the folks at SUNY are trying to deal with that question. I frankly don't know anything about the FDA approval process, but it's, you know, I'm retired from the federal government, so I certainly know what agencies go through when they have to field thousands of public comments and try to sort out what's the best way to go. But that's why we have these processes in place, so that that we don't make mistakes, and we have experts who are analyzing these things. So, Doug, maybe you can add something to that.
Yes, a couple of quick things. The earlier part of the comment, not only is there an annual meeting of the foundation itself, I think 2 or 3 years ago, it was actually in Portland, Maine. It's often down south, but every year the Vermont, New Hampshire chapter has their annual meeting, which everyone is invited to. In fact, this year it was going to be in Plymouth, New Hampshire, and next year I suspect it will be in Plymouth in April. Hopefully we can get together in person then. And for Kurt might have already put out this pitch, but for $40, you can join the American Chestnut Foundation, and you automatically would be joining the Vermont, New Hampshire chapter. You get 3 or 4 wonderful magazines a year, all kinds of updates. It's a good investment. And then also, if you do well, please go to the website for the transgenic approval and make a comment. I mean, it could be negative, obviously. We hope it would be positive, but you do not need any special expertise to make a comment. Anyone can make a comment. Your name gets registered. I don't know that your location does, but yeah, everyone, please do that. Thank you.
I'll add one other thing. We use Constant Contact as our email communication process, and all of our members are automatically included, those who give us an email address. But we have hundreds of other people who are not members who we've met at different meetings or events, and they simply give us their email address. So Tara, I guess you'll be putting up my email address and in your comments. So if you want your name added to the list, I'll even sneak you in there. If you don't become a member, just send me an email with your email and address, your name, and what town you live in, and I'll add you to our Constant Contact list.
Excellent. Okay, well, we have one more question, and I will again post all of this information on our website and on our Facebook page for those of you who are familiar with Green Mountain. We have a follow-up from Allison Leach, who's the person that mentioned they have the American chestnut tree in their yard. She says, yes, we are in Maine. We will be willing to share our nuts with all chapters and have a multiple trunk chestnut tree that one stem is blight ridden and the other 3 have no signs of blight. So that has been interesting to them. I am also chair of our conservation committee, and we have a chestnut introduction plan in place, and we'll be looking into contacting some new people. So that sounds very promising. So maybe Allison can reach out to you and Doug as well to share those nuts with other chapters.
Wonderful.
All right, so I think we're gonna wrap up. There are no other questions here, no other comments to share. I wanna thank you, Kurt, for your wonderful presentation, very informative and interesting. And I hope folks do end up reaching out to you and going to the American Chestnut Foundation website, learning more, joining as a member, and then following up, maybe planting a sapling at some point. And sharing your comments on the website there about the transgenic patent and helping to promote their cause. And thank you also, Doug, for joining us and helping to answer some of the questions tonight. We really appreciate all of your work in conservation and wish you the best at the American Chestnut Foundation.
Thank you.
Again.
Mark, thanks for making this happen.
Yeah,
Absolutely.
my pleasure. That was great.
Very good. Thank you all.
Do you have any
All
idea
right.
how many people?
Oh, Tara, are you still there?
I am.
Do you have any idea how many people joined us this evening?
We had 23.
23. Okay.
Yep.
Very
No
good.
different than doing it.
Thank you.
No
Thank you
different.
for,
We went to
All
the
right.
library, so it wasn't a huge.
Bye.
Your background, Kathy. Nicely done, whatever your daughter. I'm done.